Okay, guys and girls, so today's going to be, I think, two videos that we're going to be uploading, and the first topic we're going to be looking at, and then all of B2 will be done, by the way. So the first topic we're going to be looking at is genetic engineering. Okay, so oops, too high. Genetic engineering. Engineering. Okay, so um, basically this is to do with the production of insulin, and obviously, as you should know from B1, insulin is needed for diabetics. Okay, uh, now before in the past, before this genetic engineering, it used to be taken from dead cattle, that's cows, or dead pigs. Now, this there's kind of a few negative points that you need to be aware of. Number one is that people who are vegan, cough cough, we all know who we're talking about, or some people with religious views. So some people may say, well, no, pigs are not allowed, or you can't kill animals based on a religious view. Um, these people are going to have an objection to using animal insulin. Now, the third thing is that animal insulin is for animals, right? This is a common vegan uh, discussion point as well, as we all know, because we all know our vegan friend. This is for animals, and it can cause side effects or uh, a response to your body. Okay, so your body could end up rejecting it and causing some side effects, and so it's not ideal. However, back in the day, you had no choice because obviously, you know, if you needed your insulin and you were diabetic, you're gonna have to have it. So it caused some problems. But nowadays, though, we can use bacteria in order to um, to make insulin. Okay, so how's that gonna happen? Now, first, what we're gonna do is we're gonna look at bacteria cells themselves. Okay, so we're gonna look at the structure and function of bacteria cells. So here's my bacteria cell that I'm drawing. Now, as you know, bacteria are prokaryotes, right? So if they are prokaryote, they're not going to have a nucleus. So they've got this free-floating chromosomal DNA, and they've got this ring of plasmid DNA. Okay, so what you need to be aware of are the function and structure of bacteria cell stuff. Okay, so number one, if we label this the cell wall, let's use a different color one. So this here is my cell wall on the outside, and it's going to be used for protection and provide structure. Protection and structure. Next, on the inside, we're going to have the cell membrane. So here's my cell membrane, and this is, of course, just like the door, controls what goes in and out of a cell. I'm just going to write it in note form, so controls what goes in and out of a cell. Here is the chromosomal DNA. Okay, so this is the DNA which is going to code for proteins and make stuff within the bacteria cell. So whatever proteins it might need, whatever enzymes, going to make that. And guys, I just want to emphasize again, bacteria, prokaryotes do not have a nucleus. So this is chromosomal DNA, and it's free floating DNA, chromosomal DNA, and it's free floating. It is not in a nucleus. Now this bit here, this is the plasmid DNA. Okay, and your textbook describes this as extra DNA, and we're going to use this DNA, this plasmid DNA, for genetic engineering. We'll cover that in a bit. And this here is a flagellum, which is basically can be used for swimming and moving around from place to place. Okay, so that is the anatomy, if you like, of a bacterial cell. Now you can have more than one plasmid cell. However, your chromosomal cell is your chromosomal DNA is your chromosomal DNA. Only one of those. So let's have a look now at genetic engineering. Okay, so what you're gonna do is you're gonna take the plasmid DNA. So you're gonna have a bacteria, right? So here's my cell wall, my cell membrane, my flagellum, my chromosomal DNA, which is free floating, my plasmid. So number one, what you're gonna do is you're gonna take your plasmid DNA. Okay, so we're gonna remove the plasmid DNA, So remove the plasmid DNA, and then what we're going to do is number 2, we're going to cut the DNA. So if this is my plasmid DNA, I'm going to use cutting enzymes. I'm going to use cutting enzymes to cut the plasmid DNA. So that's number 2. So there's my cutting line. I cut the plasmid DNA, pDNA, using cutting enzymes. Nicely named, isn't it? I think so. Because if it's called anything else, you'd have something else to remember. So we've got our plasmid DNA, we've cut it open. Then what we're going to do is we're going to take some some chromosomes that code for human insulin. So here's my human insulin DNA, human insulin chromosomes. Okay, so this is going to code for human insulin. So what you're going to do is you're going to have, so that's number 3. So number 4, what you're going to end up having is you're going to end up having this plasmid DNA, which has been cut, plus your human insulin chromosome. And what you're going to do is you're going to stick them together using sticking enzyme. So let me just go back to the original color of the plasmid. So you're going to have this, and it's going to be stuck with your human insulin chromosome. And you're going to stick these together. So stick the pDNA plus the human insulin chromosome. I'm just going to write it in short. Using sticking enzyme, another nicely named thingamajig. Using sticking enzymes. Right. Then what you're going to do is you're going to place this newly attached, this new plasmid DNA which has human insulin back into the bacteria. So the bacteria is now going to have this thing here. So let me just draw that in. So it's gonna have this plus the human insulin bit. Once it has that, the bacteria. So that's point number 5. Really, is just sticking that back into the bacteria. Point number 6. Let's change color. Point number 6 is going to be. Let's change it again. Still. Point number 6 is that the bacteria will divide by mitosis. Will reproduce / divide, same thing, by mitosis. Now why is mitosis an advantage here? Because it's going to produce genetically identical copies of itself, because that's what mitosis is, genetically identical copies. And so what that means is that you're going to have loads and 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 loads of human insulin being produced. And so that's really good, isn't it? Because that's what you want for these diabetic patients. So human insulin being produced, quite a lot of it. And so now, number 1, if you look back at the problems that we had, we've eliminated all those problems. Some people who have these religious issues about taking perhaps pork insulin or cattle insulin, that problem's gone. Your vegan problem is gone. And also here you got lower chance of rejection or reaction from the body. And why does this lower the chance? It's because it's made from human genes and it's coded for by human chromosomes, and so you're not going to have this problem. So there's a lower chance of rejection from the body and a lower chance of reactions. However, occasionally mitosis might go a little bit differently, so they might replicate a little bit differently, and you may have some insulin that's produced that doesn't copy 100% properly, and so you might have still a small problem. So you do need to know about that, guys, that occasionally you've got a small problem in terms of mitosis and the way it replicates. And so sometimes they can be, but the chances is way smaller compared to what we used to do in terms of animal activity. So I hope that clears that. So that's bacterial cells done. I'm going to upload this video and then we'll do another one on another topic.